In this session, we'll be looking at the use of reporter genes, and it's nicely fitting with the previous presentation about the aptamers, because there's a challenge, as we previously heard in the previous talk. Nucleic acids don't penetrate into the cells, they don't cross the cell membrane, so if we want to introduce reporter genes into cells, we'll have to use some tricks, and I'm going to try to explain you what the available tricks are at the moment in September 2013. Disclosures, we're not going to spend any time on this. I just want to stress out that this is part of a 3-year educational program, and the reporter gene systems as such have been explained by my former mentor, Professor Bakeland. I will talk about the introduction of the reporter genes, and specifically for reporter gene animals, we'll have to go to Seoul next year if you want to hear more about this. Now, as I already mentioned, we have a toolbox at our disposition, and this ranges from some very simple and basic lab techniques that most of us have all done, going from transient transfection to the use of more sophisticated viral vectors that I will explain into detail. And then more recently, and very exciting, we have new site-specific genome editing tools at our disposition, and I think that these can become very interesting for the use of reporter genes in the future, and we will discuss those also in detail. Just a brief summary, what is a reporter gene? A reporter gene is basically a stretch of DNA which after translation to RNA, mRNA is translated to a protein, and this protein can then easily be detected, not only be detected but also be quantified. And there are many different types of reporter genes. So the early years were colorimetric assays, luminometric assays, in situ and fluorescent. Proteins are now the mainstay of biological work. Then, of course, we wouldn't be at an imaging congress if we wouldn't talk about imaging reporter genes. And these have the specificity that they can be detected in vivo through non-invasive techniques. So the proteins are responsible for a physical signal that can be detected by imaging hardware, can be a bioluminescence camera, can be a PET camera picking up a radionuclide which is decaying, binding, or after modification by the protein. Of course, there's also MR and fluorescence, you know all of this. Now, this is not the answer to the question that I received from the organizers, but it's still something interesting to think of, is what are we going to link the imaging gene to? In gene therapy trials, you often want to introduce a therapeutic gene or a gene of interest, and you want to link a reporter gene to be able to see how your gene therapy strategy is working. And there are a number of different ways how you can actually couple your reporter gene to your gene of interest. The yellow stuff that I use over here are promoters. The green represents your gene of interest or therapeutic gene, and the red is the reporter gene. And you can simply use two constructs. So you actually do the work twice, and you transduce or transfect twice your cells. You can make one large fusion construct in which you have your two promoters driving both of the genes. You can go for a fusion protein. I'll show a small example of this, where you actually make one fusion gene that will then result into one fusion protein. But this, of course, if you're using a therapeutic gene, you're going to modify your therapeutic protein, and this might have consequences and not be readily translatable to the clinic. If it's just for reported genes, such as HSV-TK or luciferases or fluorescent proteins, it can be okay. Then you can, what is often used are IRES, internal ribosome entry sequences, which is a small patch of DNA which is in between your two genes, and in the subsequent RNA you will have ribosomes that will start binding at this site and then lead to translation of the more distal encoded gene. But typically the distal gene is expressed at a lower level than the more proximal gene. You can use bidirectional promoters, which will induce translation of the two genes at one session, but typically they tend to be somewhat weaker, and there can be asymmetrical effects in that one direction is transcribed more than the other direction. And finally, you can use a system consisting of peptides which are derived from picoRNA viruses. As you know, viruses have a small genome, so they need to encode a lot of information with a limited amount of space. And these are actually fragments that, when the ribosome will pass on the corresponding RNA, will introduce a link, a niche in the polypeptide chain. And so you will just get different independent proteins that are being expressed, but they will have a small tag consisting of this peptide. And this has been demonstrated for gene therapy trials that, for instance, you can express in a stoichiometric manner, so in a 1-to-1 ratio, up to 4 different proteins at once. And in our lab, we have been using these peptide-2A sequences to generate triple fusion reporter probes encoding fluorescent, bioluminescent, and PET reported genes that are then each expressed in an independent manner. So you don't get a fusion protein, you get 3 independent proteins. Now, this is the first part of the answer to the real question I got from the organizer. So how do we get the genes now into the cells? And the most simple way of doing this is by transfection. And so the basic problem is that your DNA is actually negatively charged, and by using lipid-based molecules that have a positively charge, you can actually neutralize the charge of the DNA, and then this can cross the phospholipid membrane, and then the DNA is unpacked and can then go into the nucleus, where 2 things can happen. You will either get transient transgene expression from episomal DNA, or you can get, in a small fraction of the cells in which you were able to introduce this DNA, you can get integration, and this will lead to stable gene expression. Now, if you look at the pros and cons of this approach, there's no size constraint. This is very low cost. This is easy to do. It's rapid. You get the results by the next day. Just give it 24 hours to transcribe and translate. You get high expression levels, very high expression levels with transient transfection, and it's non-immunogenic. On the contra side for transient transfection is that you do get supraphysiological expression, which, if you're studying the function of specific proteins, can be a problem. But for reporter genes, typically the more of expression you have, the better you will be able to pick up the signal. So this is not a problem. So this is for transient transfection, where the DNA maintains episomal. Now, typically, as I mentioned, a small fraction of the cells, you will be able to get stable integration, and typically by adding a selection marker, an antibiotic resistance marker, for instance, you can then add the antibiotic, and you can select these cells through Darwinian selection. And if we look then at stable transfection, you have the same pros, that it's easy, low cost, non-immunogenic, and you can put up constructs of very large size. The contrast is that it's more time consuming, because now you need to do this selection step, which can take weeks to months. The expression level is going to be variable, but it's definitely going to be lower than with transient transfection, where you get a huge boost of the episomal DNA. Now, this episomal DNA has now been degraded by the time you have selected these cells, and so the expression level is going to be much lower. You will have to perform clone selections. You will start with specific clones in which the integration event took place. And this is something important I'd like you to remember. The integration which is occurring is occurring in a random fashion. You don't know which chromosome has been targeted. You don't know where it is. You don't know if it's in a stretch of DNA which is going to be modified during specific processes. For instance, during maturation or differentiation of your cell, you don't know where this is going on. So now I'm going to switch to viral vectors that have been developed. Now, viral vectors, when you introduce DNA into a cell, this phenomenon is called transduction, and transduction is different from infection. Infection, which is occurring with viruses by themselves. So I'm going to first explain what an infection is doing. So you have a virus. This is an RNA virus. It gets into the cell, liberates the capsid, and then the RNA, which is to RT transcribed into DNA. A pre-integration complex is made, and then this is integrated into the DNA. And then from the DNA of the host cell, we get production of novel virants that are then capable of leaving the cell and then going to other cells. And so you get a spread from one cell, the primary affected cell, to other cells. You get second and third round effect. Now, the difference with a viral vector is that it's engineered so that it does not contain specific elements, and so that once the element is integrated, you do not get any formation of new virants anymore. You only get expression of the transgene that you have chosen to introduce into this viral vector. So there are no new virants being created. It's a single round phenomenon, and each virant that you have produced will target only one cell in your experiment, but of course one cell can be targeted by many different virants. So the first category that I'd like to explain are the adenoviral vectors. These are DNA vectors which can carry a quite high payload of up to 30 to 35 kilobases, and after introduction to the vector, they will deliver the DNA into the cell, and it will stay as episomal DNA in the nucleus and then lead to production of your transgenes. The advantages of adenoviral vectors is that they can carry a high payload, so you can introduce large genes, quite easy to be produced, and they can transduce dividing and non-dividing cell. And this is a distinction that I will make for a number of different viral vectors. So it's important with adenoviral vectors, you can target both dividing and non-dividing cells. On the contra side is that they are immunogenic, and that because it's only episomal, you will have short-term expression. So this is typically, if you do this, it's not often used in cells, it's more often used in animals, but your expression is typically indetectable by 2 weeks after injection of the adenoviral vector. So this is only for short range. This is just one example of detection through PET using the HSV-TK system of adenoviral vectors that were injected into liver metastases in a patient, and we can clearly see the FHBG which is taken up by the metastases in these cells. So it is something which is translatable to the clinic, and here you see that if you inject more viral particles, 10 to the 12th, compared to 2 times 10 to the 11th, you get more signal in the liver metastases. I then move on to adeno-associated viral vectors. These are also DNA-based vectors, but these can only carry a much smaller payload. It needs to be smaller than 4.5 kilobases, so almost 10 times as small as for the adenoviral vectors. It's a single-stranded DNA vector, and so these double-stranded DNAs will go to the nucleus, and there they can either stay episomal and lead to transgene expression, but they can also integrate typically on chromosome 19, and they can thus lead to long-term stable gene expression. The pros is that it's non-immunogenic. It's a low-risk type of viral vector because there's no human pathology associated with the adeno-associated viruses, and you can target both dividing and non-dividing cells. On the contra, the insert site is going to be low. For imaging reported genes, this is not a problem as such, but if you want to hook this up with some therapeutic genes or other gene constructs, you might run out of space. And the production process is more complicated than for adenoviral vectors. You will need some people that have skills in the production and the purification of these adenoviral vectors. Now I'm switching gear to the retroviral vectors, which are RNA-based vectors, of which the family of which HIV-1 is a known virus, and these are only capable of transducing dividing cells. So for the process that I'm going to explain now, the cells that are targeted need to go to mitotic phase. And this is certainly a drawback of this vector system. If you want to go in vivo, if you have cells in culture and they are growing, then you can use them, and this is why these are often used in cell culture experiments. So it's an RNA-based viral vector. The payload is less than 8 kilobases, so this allows you to introduce a number of gene constructs, and for instance, you can easily introduce GFP with an iris and a thiophyluciferase construct. You will then get the RT step that I already explained previously, and the integration into DNA, and from that you will get a stable transgene expression. Now the main advantage of this system is that you can get long-term expression, so you do get integration of your transgene of choice into the genome of your cells. The drawbacks is that, as already mentioned, you can only transduce dividing cells. The production process is more complex, so you will definitely need some expert help over there. You can activate some genes because the insertion of the cassette will be at a random site, and by going into specific sites, you can augment or diminish the action of endogenous genes of your cell, and this can lead to a so-called insertional mutagenesis, where the cells, for instance, starts to behave in a cancerous way. One thing that's also very important to realize is by using this strategy is that your cells are non-isogenic, for which I mean that if you start by transducing a pool of, let's say, 1 million cells in vitro, that each and every single of these cells will be targeted at a different integration site. So the integration sites will be different for every cell that you have in your mixture, and also the number of inserts will be different. If you put a high amount of these vectors onto your cells, you will have cells that have 1, 2, 3, 4, and many integration sites. So you end up not with a homogeneous cell population, but actually a mixture of a very large number of different sites. And finally, it's known that the areas that are targeted by these retroviral vectors during differentiation, for instance, can be silenced. So you can then lose your transgene expression, and so you lose the detectability with the reported genes. And for this, I would like to switch to the lentiviral vectors, which are actually a subfamily of retroviral vectors that are responsible for slow disease processes, and HIV is actually one of the lentiviral vectors. And these can also transduce now non-dividing cells. HIV is lymphotrophic. These cells don't divide when they get transduced. So this is a major step forward with the use of lentiviral vectors compared to retroviral vectors. The payload is similar to retroviral vectors, and also the integration process is similar. So on the pro side, we have long-term gene expression, and we can use this for dividing and non-dividing cells. And for instance, you can target neurons in vivo by the use of lentiviral vectors, which you cannot do with retroviral vectors. And on the contra, you have the same weaknesses as for the retroviral vectors, except of course for the non-dividing cells. And this is just one example where we used triple reporter gene with fluorescence, luciferase, and HSVTK mutant. We produced a lentiviral vector with encoding this protein. We transduced the cells. We could see this by fluorescence microscopy, detect with BLI, see it with micropet, and then we could start using these to generate metastatic mouse models where we injected our melanoma cells. After a while, we get metastases, and we could try to interrogate where the cells were by using PETs over different time points, or by performing bioluminescence to see them, and localize with CT to find the cells. So these were all approaches that are either easy to perform and are part of basic lab science, such as transfection, and then the more classical agents that have been developed to introduce cells into genes. Now I'm going to move forward to a more novel area in this field, which is site-specific genome editing. And there are actually two systems of so-called nucleases, which are enzymes that are going to cut DNA, of which one is the so-called zinc finger nucleases, which has been around for quite a long time, almost a decade. And the other one is a so-called transcription activator-like effector nucleases, but everyone calls them TALENs, which are basically proteins which will bind to DNA. Now, we heard from the previous talk that DNA can bind to a number of structures, for instance, to proteins, but it's also the other way around. Proteins can bind to a DNA, for instance, in transcription factors. And the nice thing about these zinc finger nucleases, or these TALENs, is that they can be not only bind to specific sites of DNA, specific stretches, but they can also be engineered to bind to the specific site or stretch that you're interested in. And this has opened the area to use these to introduce genes at specific sites of the genome, sites that you can choose. And so the system of how this works, and this is roughly the same for zinc finger nucleases and for TALEN, so I will explain them at the same time, is that you have a DNA binding domain, which is of protein nature and which has been engineered to bind to a specific site. To this is attached a FOC1 nuclease, and then you, oops, a little bit too early, you make a so-called donor DNA plasmid in which you put your construct and then you put sites of which are homologous to the site where in the genome of your host cell you are interested in of introducing your construct. And then by performing transfection, you introduce this material into the DNA of the cell, and then the nuclease will actually introduce a cut, and by engineering your DNA binding domains so that they are complementary, you will engineer not just one DNA cut, but you will actually introduce a double strand break, and then through homologous recombination, your construct will be inserted because of the homology elements into the DNA of your host genome. Now, this strategy has as main advantage, of course, that it is site-specific. So you're not just randomly putting something into the genome of your cell, but you can specifically choose a site where you want to introduce this. And not just for reporter genes, but for medical applications, therapeutic applications, you could, of course, target the site where a specific mutation is present. This produces isogenic cell lines, so all the cells that you have actually produced carry the insert at the same site. You don't have any mutation problems anymore if you target this to a safe site, a site where you know that there are no genes that you're going to disturb by putting in your cassette, and it can be rendered highly efficient, but that's still with people that have some experience. On the contrast side, you can introduce to a certain amount off-target double strand breaks, which are, of course, pathogenic for the cells. As you do get one cell that you then have to select and grow up to get more cells than the initial cell you targeted, you go through a genetic bottleneck and you can get some founder mutations if some mutations are introduced in the population of cells that you're then expanding. Then the one major thing I would like you to remember that it's quite complex to target a different site than a site that somebody has not targeted before you, because this is based on a protein to DNA interaction. Your zinc finger nuclease needs to recognize a specific stretch of DNA. There are companies that are expert in generating these zinc finger nucleases, but this is something which is not straightforward to do. And so because of this, your timing-wise, it will also take you at least a number of months before you will be able to have cells with your gene of interest in there. And by end 2012, Joe Wu at Stanford has published a very nice paper where they showed that indeed it was possible through the use of zinc finger nucleases to introduce reported genes, luciferases, HSVTK into cells. And so here they show the scheme again. First you introduce the cells, then you can then analyze if you didn't change the phenotype of your cells, looking at pluripotency for stem cells. And then this is the targeting approach they use. They use the site that I will explain later on. And then by PCR, they were able to show that in different cell clones, they were able to introduce the reported gene cassette. And this is a Southern blot showing that indeed they have DNA integration of the site. And then they could use this in embryonic stem cells to image the formation of teratomas through BLI and quantify the signal of the growth of the embryonic stem cells. But they could also use this to differentiate the embryonic stem cells, and then they could use cardiomyocytes or endothelial cells that they could then follow up through BLI within these mice. We have actually also been doing a similar work, but we've been using different PET reporter genes, and the site that we are targeting is the same one, which is a classical site within the field. It's the AAVS1 locus, which is encoding a phosphatase, of which name is seen over here. It's on chromosome 19, and it's known to be a so-called safe harbor because there are no genes that are in the vicinity that you're going to interrupt by putting, for instance, a strong promoter over there. And it's also a site which is not epigenomically silenced when, for instance, stem cells are differentiating, so that you will not lose the expression of your reporter gene when, for instance, you're differentiating stem cells, as I previously showed on the work from Joe Wu. And here you see our construct. So we have been using our T2A, our peptide 2A approach. So we introduce a gene for both EGFP, 5'-luciferase, and then 2 different PET reporter genes. We were able to show that our cells, well, are nicely fluorescent, as you can see from the slides. The cells are bioluminescent, so the luciferase is active on there. This is quantification of the signal. Controls are somewhere around here, so we have 2 orders of magnitude higher. And this is the radio tracer experiment showing that we have a very high uptake in the cells of the radio label compared to the control cells. So we were able to reproduce in that indeed you can use zinc finger nucleases to introduce reporter genes into stem cells. And now, finally, if you're still with me, if you're not anymore, you need to wake up, because this is something which is very important, which is very exciting, and you should take note of it. This is a novel system which has recently been discovered. It's already out there for a little while, but the fact that it's actually a very useful tool, and maybe by far the most useful of all the ones that I've shown up until now, has actually only been recently being discovered. And so it's called the Crispr system, and Crispr is actually a nice acronym for clustered regularly interspaced short palindromic repeats. And so this is something which was actually discovered first in bacteria and archaea, and it's a stretch of non-coding DNA, which previously was called junk DNA, but if something in biology is called junk, you should take attention to it, because it's something very interesting that we haven't found what it is doing yet. And so recently this has been unraveled. So how does this present? So it's basically, it's a palindrome where you have one sequence of DNA and you have the other sequence in reverse, which is then repeated at short intervals. So this explains the palindromic repeats, and these are also short. And then in between of them you have a spacer, which is typically around 30 base pair, and this is the regularly interspaced, which is expressed, and they can be present at in many repeats, so which explains the cluster that you have over there. And these things are actually present in up to 40% of bacteria and 90% of archaea, which suggests that they have an interesting role to do. And things gotten into accelerated when people realized that the spacer DNA actually has homology to phage sequences. And it turns out that actually that this system is a DNA-based type of immune system which is protecting bacteria and archaea against viruses, the phages that are infecting these cells. And yeah, so this should be over here. I already explained this. So what happens is that this stretch of DNA, when there is a phage infection, it gets translated into RNA. And so you get then this molecule, and then these palindromes, they fold up, and then you get this folded RNA molecule. And then this molecule associates with an enzyme called Cas9 for CRISPR associated, Cas9, which is actually a nuclease, so again a DNA cutting enzyme, but this is actually a nuclease that will cut double-stranded DNA at 2 sites, so it's actually a double strand break inducing nuclease. And the RNA which is introduced with within the enzyme is called the CR from CRISPR RNA. And so as I mentioned, it's a nuclease, has 2 active cutting sites, 1 for each strand. And so how can this then be used to introduce reporter genes into the cell? Well, you use the Cas9, and then you engineer a stretch of RNA, of which this part of the RNA is actually complementary to the site of your locus that you want to target into your genome. And again, you use a donor DNA plasmid with your construct and then your homology elements, and then you transfect this again into the cell, and then we'll have a little look at what happens. So what you have is that the RNA, which is in your Cas9 enzyme, will bind to the site of DNA that you've chosen to target, and then the nuclease activity will introduce again a double strand break, and then again by homologous recombination you will get introduction of your construct into the DNA. Now, for those who said, but isn't that the same thing that you explained with the zinc finger nucleases and the tail ends? Well, no, the main difference here is that the targeting is done by an RNA structure, and it's much easier to generate an RNA structure which is complementary to a specific DNA sequence. This is just every day-to-day molecular biology, than it is to engineer a protein which is complementary to a DNA structure. And that's the reason why this is getting a lot of traction for the moment. And this is just one paper from March 2013, where they used the Cas9 system, CRISPR mediated, just to introduce cassette into the genome of human cells. And here you see that they were able to get this in, and with the proper controls you don't get anything. And they were able to express GFP. Now I know that GFP is definitely not the reporter gene of choice for molecular imaging, but this was a proof of principle paper. And there have been many other papers that are either trying to optimize this system or to try to use it for many different applications. And with this system, you're really getting to gene surgery that we could use to correct specific sites of DNA. And so the pros are, of course, that it's site specific. You do generate isogenic cell lines. It is safe. It is highly efficient. Main advantage compared to the zinc finger nucleases and talents is that it's RNA-based locus targeting, so you don't need to engineer proteins that will bind to a specific stretch of DNA. And you can also even target multiple loci at the same time just by using different RNA guidance molecules. On the low side, there might be some off-target double-strand breaks, so this is definitely one of the areas where this CRISPR system is being optimized as we speak. We do also have genetic bottlenecks if you have to select your cells out after you have introduced the modification, and then it will take you, again, a number of months before you have the gene into the cell. And so I'd like to summarize with the different techniques that I've mentioned. So we do have transfection, which on the pro side is cheap and easy. You can insert large construct and it's non-immunogenic. You do get non-isogenic cell lines, and you cannot use this ex vivo. And there are differences for transient and stable transfection. Transient, well, it's high efficient, high expression, it's rapid. For stable transfection, it's easy to do, but you do get low expression rates, and you have random integration, and it's also slow. For the viral vectors, well, they are very good at doing what they do, introducing genes into cells. Viruses have been evolved over millions of years through nature to do exactly this, so they are efficient. When you use viral vectors, you only have a limited cell selection to do, because most of your cells will be target if you use appropriate titers. And this is something you can also use in vivo, and this is something where the genome editing systems are currently not ready for in vivo use yet. So I still think they are the best choice for in vivo insertion of cells of genes into cells. On the contra side, you have biosafety issues, and you can get multiple transductions per cell. There are differences in between the different viral vectors. The adenoviral vectors, they have short-term expression mainly. Adeno-associated viral vectors only low payloads, but you can get long-time expression. Retroviral vectors, again, long-term expression, but you have problems of non-isogenic cell lines, insertional mutagenesis, and silencing. And the same goes for the lentiviral vectors, but they have the advantage that they can target both dividing and non-dividing cells. And finally, we have the site-specific genome editing strategies, which have the advantage of being safe, site-specific. They do generate isogenic cell lines. There is no silencing if you target a safe harbor locus. On the contra sides, you can get genetic bottlenecks. Off-target double-strand breaks is still an issue. It will take you much longer than with transient transfection or by putting on a lentiviral vector if you can get someone from somebody else. It's only ex vivo use for the moment, and yeah, for some of these techniques the experience is limited, although this will rapidly expand. And then specifically for zinc finger nucleases, it's difficult to design zinc finger, the zinc finger part of it, so that requires specific expertise. With the TALEN, this is easier to do, but I still think that the Crispr system, where you can use RNA-DNA hybridization, is actually a very nice alternative for the system, and this can also be used to simultaneously edit multiple target loci. This is some further reading for those who would be interested, and in the manner, these are some of my co-workers I would like to acknowledge, and then finally, if you have any questions, I would be happy to take these. Thank you very much.